Great, and our next speaker is Mark Herman from Duke, who will be speaking on fructose metabolism and insulin resistance.
Thank you, Dave. Great. It's a pleasure to be back in Boston. I feel like I was here only 2 years ago and moved to Duke relatively recently. So it's been great to see friends back here. So I'm going to talk to you today, and I should say I should thank Kimber Stanhope for a fantastic talk and really an introduction to a lot of the topics I'm going to talk about today, which deals with fructose metabolism and insulin resistance and other cardiometabolic features from a molecular mechanistic perspective. But before I get into that, this is some work that published recently in collaboration with Nicola McCowan, who's a nutritional epidemiologist here at Tufts, looking in, this is the charge consortium, looking to see whether sugar-sweetened beverages affect circulating insulin levels at levels ingested at typical levels in the population. And we did see that among these 35,000 adults, there is a dose-dependent increase in circulating insulin levels associated with sugar-sweetened beverage consumption. And this is some unpublished work within the Framingham cohort conducted by Daniel Haslam, who is also here today, looking at dose dependence of sugar-sweetened beverages. This is again adjusted for total energy intake on HDL cholesterol going up here on the right, and triglycerides going up here on the right, and HDL cholesterol going down with increased sugar-sweetened beverage on the left. And these were really pronounced dose-dependent effects within a relatively small population, again, within the range of sugar-sweetened beverage commonly ingested by people in their daily routine. But as has been discussed by numerous presenters today, in particular Frank Hugh, there are some caveats when discussing the relationship between sugar or sugar-sweetened beverage with cardiometabolic risk factors. Particularly that sugar-sweetened beverage associates very well with these risks, but other measures of dietary exposure are less consistent. For instance, fruit, as indicated by Dr. Ludwig recently, just a few minutes ago, may be protective when on these factors. So from my lab works in molecular mechanisms by which nutrients are sensed and by which they regulate metabolic processes, and we hope by better understanding these mechanisms by which sugar consumption is sensed and regulates metabolic programs, that we may be able to understand how these adverse effects develop, and maybe be able to realize some new answers to unravel these epidemiological controversies. So when we talk about sugar in my laboratory, we're mainly referring to sucrose, which is, again, one molecule of glucose and a molecule of fructose. High fructose corn syrup, which has been discussed, again, is about equal portions of glucose and fructose, though predominantly in the monomeric forms. And it's long been described in literature, in rodent studies, that when rodents are provided large excesses of fructose in particular, you can rapidly develop all the features of metabolic syndrome: obesity, hypertension, insulin resistance, fatty liver disease, increased uric acid, within a matter of weeks. Now, the doses used in these studies are far in excess of what is commonly ingested by people. Nevertheless, it shows that fructose itself in high quantities can reproduce all of these features, and if we can understand how that's happening, maybe we can provide some insight into how to better treat people. So this motivates our laboratory. Can a detailed knowledge of the biology of fructose metabolism help us understand the pathogenesis of insulin resistance, dyslipidemia, and cardiometabolic disease? Can it help us resolve outstanding questions and controversies related to the public health effects of sugar consumption? Kimber Stanhope discussed this a little bit about the differences between fructose and glucose metabolism. They're not biochemically, they're not metabolized the same way. So fructose is transported into cells, whether this is enterocytes or liver cells, by a number of different sugar transporters, and phosphorylated by this enzyme, ketohexokinase or fructokinase, to this metabolite fructose 1-phosphate. So fructose 1-phosphate is a metabolite that's unique for fructose metabolism. It's not produced in the glycolytic or gluconeogenic paths. In contrast, glucose is phosphorylated by this enzyme, glucokinase. Now you can see the KMs for these enzymes. The KM for glucose of glucokinase is about 10 millimolar, and the KM for fructose or fructose kinase is 0.8 millimolar. So the affinity of ketohexokinase for fructose is much, much higher than the affinity for glucokinase is for glucose. And this is one of the reasons, one of the number of the reasons that fructose can be metabolized so quickly in cells that express this enzyme. So when fructose is metabolized, phosphorylated fructose 1-phosphate, it's cleaved by this enzyme called aldolase B to glyceraldehyde and dihydroxyacetone phosphate, and this is then phosphorylated to glyceraldehyde 3-phosphate. And these are metabolites within what we call the triose phosphate pool, the 3-carbon phosphates. They're common for glycolysis and gluconeogenesis, so they mix with all of the metabolites in the glycolytic and gluconeogenic pathways, and they can be metabolized back up to glucose 6-phosphate and made into glucose via glucose 6-phosphatase, the rate limiting or the rate determining step in glucose production, or they could be metabolized to pyruvate and lactate and used in the TCA cycle or exported as lactate from cells. So Kimber mentioned one mechanism that distinguishes how much fructose versus glucose can be metabolized in the liver, which is the phosphofructokinase step. But there's actually another more proximal step that is really important for determining how much glucose can be taken up in the liver in contrast with fructose. And that's the fact that in the liver, unique to the liver, glucokinase, again, this enzyme that phosphorylates glucose 6-phosphate, is actually held in the nucleus in an inhibitory state by this protein called GCKR, glucokinase regulatory protein. So this is a very, very tight interaction in the liver, and the vast majority of glucokinase in a fasted person or animal is actually in an inhibitory state. So even if you gave that animal glucose, it's not going to get phosphorylated, it's not going to enter the liver and get phosphorylated because there is very little active glucokinase around. But it turns out that this interaction is destabilized by fructose 1-phosphate, this metabolite that is specific for fructose metabolism. So fructose 1-phosphate destabilizes this interaction, allows glucokinase to exit the nucleus to enter the cytosol, where glucose can now be phosphorylated to glucose 6-phosphate and utilized by the cell. So if you think about this, this is true in people, this is true in mice and rats and every animal that's been studied. This is actually probably an evolved mechanism. So the liver can tell when there's a lot of glucose 6-phosphate around, did it come from sugar that I just consumed, or is it coming from a gluconeogenic process where glucose 6-phosphate is being made to make glucose? So by using F1P as a sensor for this interaction, the liver is only going to take up glucose when you've ingested sugar containing fructose. And this is really important because, again, we're in an overnight fasted person or animal, when you ingest glucose, the vast majority of it actually bypasses the liver. The liver only takes up about 10% of it, and the vast majority of it reaches your peripheral circulation, stimulates insulin secretion, which allows its disposal into peripheral tissues. And that's actually why a glucose tolerance test is useful as an index of peripheral insulin resistance, because the glucose bypasses the liver and gets to the rest of the body. You need that insulin to dispose of it. But in contrast, fructose, when you ingest a load of fructose, so this is 12 grams of fructose, that fructose is transported into the enterocyte. And there's actually abundant ketohexokinase in the enterocyte itself. So ketohexokinase, this enzyme that phosphorylates fructose, expressed highly in the intestine, the small intestine, the liver, and also the proximal tubule of the kidney. And we don't really understand much about what it's doing there, but that's probably very interesting too. But a large portion of ingested fructose may be getting metabolized within the enterocyte itself, and some fraction of that, and that probably depends on the load of fructose, gets past the enterocyte into the portal vein and transported to the liver. So when you give an oral fructose load, say 12 grams of fructose, you can measure about 1 millimolar fructose levels in the portal vein. At the same time, you can only measure about 100 micromolar or 0.1 millimolar of fructose in the peripheral circulation, which tells you that the liver is extremely efficient at extracting this ingested fructose. About 90% of the fructose is cleared first pass through the liver. And again, it's phosphorylated by ketohexokinase and can be used, can be converted to all these different substrates, which we'll talk about more in a little bit. And we know ketohexokinase is actually really important. So there's a human genetic condition called benign essential fructosuria. There are some rare individuals that have homozygous deletions in ketohexokinase throughout their body. And these people, they can ingest sugar with fructose in it with no problem. They absorb it, but they don't metabolize it at all and actually pee it all out in their urine. And these people, there aren't that many of them, but they eat sugar just like the rest of us, but they've never been reported to have any significant cardiometabolic disease. So they seem to be relatively protected. And this has now been replicated in mice. There are two groups, David Bonthron and Leeds made the first ketohexokinase knockout mice and showed that if you give ketohexokinase knockout mice a high fructose diet, they absorb the fructose just fine. It all ends, they pee the majority of it out in their urine, and they don't develop any obesity, any insulin resistance, any hypertriglyceridemia. And so what this shows is that fructose metabolism, fructose phosphorylation, is critical for fructose-induced disease. It's not the increased circulating of fructose itself. That doesn't cause really any problems. It has to be metabolized to cause problems. So how can it cause problems? There are a number of potential ways, and these are not these are not mutually exclusive. So the first step, again, in fructose metabolism is phosphorylation of fructose 1-phosphate. And again, this enzyme is really active. And when you give an oral fructose load, the concentrations of fructose 1-phosphate in hepatocytes can reach like 10 millimolar, 20 millimolar, which is incredibly high for a metabolite. But what that also means is you're consuming a tremendous amount of ATP in the cell, and you get so much ATP that you can actually detect in some individuals systemic phosphate depletion acutely after a fructose load. And this ATP gets converted to ADP, which results in production of AMP. And AMP allosterically activates this enzyme, adenosine deaminase, which results in purine catabolism, which is why you get increased uric acid production. So there's a number of investigators that believe it's this step, the uric acid production and or the energetic stress caused by fructose phosphorylation that contributes to metabolic disease. Another possibility is that, again, there's a tremendous amount of fructose entering that hepatocyte, getting converted to hexose and triose phosphates, which is adding substrate to the hepatocyte or to the liver cell. And that substrate can be converted into malonyl-CoA and used to synthesize lipids in de novo lipogenic process. And contribute to steatosis and or increased VLDL secretion. This hexose triose phosphate can actually be converted to glucose 6-phosphate through the gluconeogenic pathway and used to make glucose. So ingested fructose can be converted to glucose and put back out of the cell for the rest of the body. And then the third major pathway is that by which fructose consumption may cause metabolic disease is that these metabolites can act as signaling molecules. So they bind specific receptors within the cell and activate metabolic programs. And it's this third component that my laboratory focuses on. And we focus on really this molecule called carbohydrate responsive element binding protein, or CHREBP. Its official gene symbol is MLXIPL, which is unpronounceable, so I don't say it. I mean, even worse than trying to say CHREBP. So this protein looks something like this. It has a number of important domains. This is the DNA binding domain, so this is actually a transcription factor. When activated, it binds to DNA in specific loci and activates gene expression of specific targets. And it has this domain, what's called the GRACE domain, or glucose responsive activating conserved element. So this domain activates transcription by binding transcriptional complexes. But it also has this domain called the LID domain, or low glucose inhibitory domain. And this domain seems to sit right over this transactivation domain. And there is some glucose metabolite. We think it's glucose 6-phosphate. We're not entirely sure. It's a little bit controversial. that relieves this inhibition and allows this molecule, what we call CHB alpha, to become transcriptionally active. It binds to DNA as a dimer with this obligate heterodimerization partner called MLX, and the two of them, when they bind to their respective responsive elements in DNA, and when it's activated by a metabolite, cause expression of enzymes involved in lipogenesis and fructolysis, glycolysis, and so they regulate broadly metabolic programs. It turns out that this molecule is conserved throughout eukaryotes. So